Yes, I'm very excited to have been selected as a rising star for the IUIS 2025 conference. And what I thought I would do is to give like a little bit of an overview on the type of research that we're doing and what I would present at the IUIS. I think in the big picture, we are interested in the microbiome, so all these bacteria that humans have on their body and the bodies. And we're kind of interested in how these microbes modulate our immune system, particularly which antibody responses we make against that. Maybe just a little bit about myself, that you understand where I come from. So I'm originally from Austria. Did like undergrads in a town called Graz on like really molecular biology, hands-on wet lab pipetting. But then for postdocs, I was more interested in learning about bioinformatics, machine learning, realizing that nowadays we're having larger and larger data sets. So I was working there in Australia and near Tel Aviv. And since about 3 years, I'm a group leader here in Vienna at the Medical University at the Center for Cancer Research. And here I'm doing a combination of those two things. So we do like high throughput wet lab assays using automated liquid handling robots. And then we use our bioinformatics and machine learning approaches to make sense of that data. And we're primarily interested in how the microbiome modulates our immune system, how it influences different aspects of human health. So there's been a lot of data that the microbiome modulates how we metabolize food, the onset of certain cancer types, onset of autoimmune diseases. And many of those aspects are not just mediated by the presence of the microbiome, but rather of how it modulates adaptive immunity. And as a part of that, antibodies play a key role. So we have large amounts of microbiota in the gut, and those are kept at bay by immune responses. So we produce IgA in the gut and IgG in the blood that are binding to those microbes, but we don't really know what do they recognize, what are the exact species, the antigens, how do these responses change mechanistically between health and disease. Um, and the reason that we don't really have answers to these questions, to all of these questions, is that conventional methods like ELISAs or peptide arrays only allow to study hundreds or maybe thousands of antigens, but the microbiome is composed of hundred thousands to millions of antigens. So we need methods to screen this large space of antigens because there's thousands of species. Every species has thousands of genes. Every gene can be a potential antigen. So what we do is we use a novel high throughput method where we combine synthetic DNA libraries with phage display and next-generation sequencing by kind of encoding all those microbiota antigens as synthetic DNA and displaying them on phages that every phage has a different antigen on the surface. We can screen hundred thousands of variants and detect them very easily. And this approach is called phage display immunoprecipitation sequencing, PIPSE, which has been pioneered by Steve Allergis lab at the Broad Institute of MIT and Harvard. And we have like some papers describing this technology that we can use it to screen for the human microbiome. Then we have different papers showing how these responses are involved in diseases. We have papers like highlighting immune response to coronaviruses, different diseases. And what I plan to do here at IUIS is to give like a little bit of an overview of the work that we have done so far, as well as some ongoing projects related to the development of the human immune system, as well as data from a large consortium that I'm coordinating, where we look at different autoimmune diseases. And I'm very much looking forward to presenting in Vienna in August.